Have you ever really stopped to think about how a drug actually works? Or maybe why one drug works wonders, but another one has all these crazy side effects? Well, at the heart of those questions is this huge biochemical puzzle, the difference between selectivity and promiscuity. Today, we're going to dive into some really brilliant research that tackles this head-on.
So where do we even begin our investigation? Well, it starts with this huge, super important family of enzymes called protein kinases. Now, just think of these guys as the tiny little on-off switches inside every single one of our cells. I mean, they control pretty much everything: cell growth, metabolism, you name it. When the switches are working right, we're golden. But when they get stuck in the on position, that's when things can go really wrong, and it often leads to diseases like cancer.
So to figure out how these switches work, scientists found this amazing molecule called staurosporine. And the best way to think about it is like a master key. It can literally fit into the lock of and shut down almost every single protein kinase. Now, this makes it a horrible drug, right? You definitely don't want to flip every switch in your body to off, but it makes it the perfect tool for our investigation. It gives us a central mystery to solve. How in the world can one key fit so many different locks?
The answer, our very first clue, is found right where the key meets the lock. This spot is called the ATP binding site. See, every kinase needs a molecule called ATP to get its energy. So they all have this little pocket, this lock that's designed to fit ATP. And that shared feature, that's exactly why staurosporine can be a master key. It's a great mimic for ATP, so it just slots right into that common pocket.
But here's where it gets really interesting. ATP, the body's natural key, it's flexible. It kind of wiggles and adapts to the lock. Staurosporine, though, our master key, it is incredibly rigid. So when it binds, it basically forces the lock into one single fixed shape. And for our research, this is just brilliant. By using this rigid molecule, we can literally freeze the binding pocket in place, which lets us study its exact shape with amazing precision. And you have to appreciate the scale of this challenge. We're not talking about a dozen or so of these kinases. The human genome codes for over 500 of them. They all have a similar ATP binding site, that same basic lock, but they're all just a little bit different. Our mission, really, is to find the tiny, crucial differences that determine whether a drug will be a super selective scalpel or a promiscuous sledgehammer.
Now, to sort through all this complexity, you can't just look at them. You need something way more powerful. You need, like, a molecular forensics kit. And this is where the research gets really, really clever. They developed a new algorithm to analyze these binding sites in a totally new way.
So how does this algorithm, it's called Symphony, actually work? Let's break it down. First, you take the 3D structures of dozens of different kinases, all with our master key locked inside, and you digitally stack them right on top of each other. Think of it like a perfect alignment. Then you lay a virtual 3D grid over that shared binding site. For every single one of those structures, you map exactly where each atom sits on that grid. And after you've done that dozens of times, you can finally ask the computer the magic question. Which spots on the grid are almost always filled? It's kind of like laying dozens of fingerprints on top of each other to see which ridges and loops are the most common.
And this whole mapping process is the genius part. It converts that physical, three-dimensional space of the pocket into pure data, a four-dimensional matrix. It's not just logging the position as X, Y, and Z, but also the type of atom at that position. It turns a really complex biological shape into something a computer can actually read, analyze, and most importantly, find patterns in.
And running this powerful analysis led to the first huge breakthrough. For a long, long time, the thinking was that if you wanted to predict whether a drug would bind to a protein, you'd look at its amino acid sequence, its genetic recipe. But the data from this new approach, it pointed to something completely different. And here it is. The big aha moment. It turns out the amino acid sequence was a pretty lousy predictor of binding. But the three-dimensional shape of the binding pocket, that showed a really strong correlation. So the main takeaway here is huge. It's not just about the building blocks the lock is made of. It's about the precise 3D way those parts are arranged. You could have two kinases with different sequences, but if they have a similar pocket shape, they'll often bind to the same drugs.
This discovery led them to a totally new idea, the quasi-shape. So instead of trying to analyze the entire massive protein, this method just focuses on a handful of key points inside the binding pocket and measures the distances between them. What you get is a unique geometric signature, a kind of molecular fingerprint for each and every kinase's binding site. And this fingerprint, this turned out to be the real key to understanding selectivity.
Okay, so we figured out that shape is king, but what specifically about that shape is so important? The final piece of our detective story was to zoom in on that quasi-shape and find the exact measurements that tell us how tightly our master key, staurosporine, is going to bind.
So they used a powerful statistical analysis, it's called QSAR, which is basically just a way to find relationships between a molecule's structure and what it does biologically. And what they found with all that complex geometry could be boiled down to just two critical factors. First, the size of one specific amino acid called the gatekeeper residue, which, just like its name sounds, controls access to a part of the pocket. And second, the degree of closure, basically how tightly the two halves of the protein clamp down around the drug.
And right here, this is the solution to our mystery. It's beautifully simple. It turns out that when you have a larger gatekeeper residue and the lobes clamp down more tightly, you get a much more compact, snug-fitting pocket. And that snug fit creates a much stronger bond with staurosporine. These two simple geometric features are the secret to a perfect fit. It explains everything about why staurosporine binds like superglue to some kinases and barely sticks to others.
Now, all of this is way more than just a cool academic puzzle. What this discovery really gives us is a rational blueprint. It's a set of rules for how to design drugs. We can finally start to move away from just trial and error and move towards something way more intelligent and targeted.
So what does this all mean for us? It means that by understanding and actually measuring these two simple things, the gatekeeper size and the lobe closure, we can start to predict how a drug is going to behave before we even make it. We can start to design molecules that are shaped perfectly to fit only our target kinase and not the other 500 plus. And that is the key to making more selective drugs, which means more effective treatments and fewer awful side effects.
This research gives us a whole new way of looking at drug design. The principles here, analyzing shape, finding these critical geometric features, they don't just apply to kinases. So it leaves you with this really exciting thought. Now that we have a much better way to pick the locks of disease, what other doors are we about to open?